All right, you're alive, so please reduce the noise. If not, your mother will hear your voice. And please, if you've not subscribed to my channel, please, before the end of this class, subscribe. In fact, that is a C. Excellence Simplified. Mutation. Now, right. Mutation is any alteration. Mutation is any alteration to the DNA sequence of an organism. Mutation is any alteration to the DNA sequence of an organism. Any substance capable of causing mutation, is called a mutagen. Any substance capable of causing mutation is called a mutagen. Classification of mutation based on where they occur. Classification of mutation based on where they occur. Classification of mutation based on where they occur. Number 1, germline mutation. Number 2, somatic mutation. Let's take the first one. Germline mutations are mutations that occur in the sex cells. Germline mutations are mutations that occur in the sex cells and hence are inheritable, or they say are heritable, and hence are inheritable. Number 2, somatic mutations. Somatic mutations are mutations that occur in the somatic cells. Full stop. They occur after conception. In the developing embryo. That may become a baby. Are we there? So somatic mutations cannot be passed from parents to offspring. Somatic mutations cannot be passed from parents to offspring. Because traits are passed only from sperm and egg cells. Only from sperm and egg cells. What? Egg cells. Are we there? Alright, if you are done with that, just drop your pen and listen up. Hello, drop your pen and listen up. Just like I said earlier, I hate cramming. Understand? Now, we know that we have a genetic material, right? And it is the genetic material that helps the transmission of information, or that helps the transmission of character traits from parents to offspring. Is it not? Now, how many macromolecules do we have? Let me see we singing fast. How many macromolecules do we have? 4 macromolecules. If you don't know it, stand up. How many macromolecules do we have? Number one is what? Okay, we have 4. Number one is what? Carbohydrate. Number 2, proteins. Number 3, lipids. Number 4, you say what? Nucleic acid, right? Good. So those are the 4 macromolecules: carbohydrates, proteins, lipid, and nucleic acid. Good. Now, here we are looking at nucleic acid. Hello, nucleic acid. Now, nucleic acid is divided into 2, right? We have 2 types of nucleic acid. What and what? Thank you very much. RNA and what? DNA. The full meaning of RNA is what? Ribonucleic acid. Why DNA is what? Deoxyribonucleic acid. Good. Now, the DNA is what I want to talk about now. We know that the DNA, according to Watson and Crick, 1953, he said that the DNA is double-stranded, right? No, this one is not looking double-stranded. This one is stranded. So the DNA is double-stranded. So let's use something like this. Even if it's not well placed. Okay, this is better. So this is how our DNA looks like. Double-stranded, 2 strand. And 1 strand is called the what? Template strand. Or what? Or the what? It's a lie. Okay, let's look at the main names first. Template strand and what? Complementary strand, right? This one runs from 3' to 5'. 5' to 3'. Alright. Now, we'll look at it in depth when I talk about genetic code. But just know that the nucleic acid, the building block of nucleic acid is what? Nucleotides, right? And nucleotides are made up of 3 things. What, and what? Nitrogenous base, pentose sugar, and what? Phosphate group. So this will make up our what? Nucleo what? Please take note, they might not ask you this again. They might not ask you nucleotide is composed of what, but we'll use the idea in mutation. Nitrogenous base, our nucleotide is made up of nitrogenous base, pentose sugar, and what? And phosphate group. Good. Now, the nitrogenous base can either be what or what? Purine or what? Or pyrimidine. You still remember? Purine or what? Pyrimidine. And we'll give examples of purine as what? Adenine and what? Guanine. Why examples of pyrimidine as what? Cut. Some very cytosine, uracil, and what? And thymine, is it not? Is it not? Is it? Now, look at it here. This is our nucleotide. I will say the nucleotides are made up of 2 things, 3 things. Nitrogenous base. I will say the nitrogenous base can either be what or what? Purine or what? Pyrimidine. A simple way to know which is purine actually have fused rings. 2 rings. Nobody will bother you to draw in this course. Nobody will bother you to draw. But this is how your purine looks like. 2 rings fused together. And your pyrimidine is just 1 ring. It's actually called nitrogenous base because as you can see in the ring, there are nitrogens here. So this is how your purine looks like, and this is how your pyrimidine looks like. That's not our point of interest here. Now, our purine example, pure as gold. Pure as gold, purine. As is what? Adenine and what? Guanine. So we can say pure as gold. Now, pyrimidine. Examples of pyrimidines are cut, cytosine, uracil, and what? Thiamine, right? Thiamine, good. Now, one thing we should know here is uracil is only found in what? RNA, right? It's not found in DNA. Uracil is only found in RNA, while thiamine is only found in what? DNA. Other ones can be found in any of them, but uracil only RNA, thiamine only DNA. Is it nucleic acid like your DNA, your RNA? But I'm looking at the DNA, but it applies to both. What makes, sometimes you're moving on the road, they said you look like your mom. They've not seen you before. Say you look like your mom. Are you the daughter of so person? Because of resemblance, right? It means you are inheriting something from your mom or your dad that makes you to look like them. And this is transmitted in form of our nucleic acid, DNA. Now the DNA, which is one of the nucleic acid, we say the building block. Just like the building block of protein is what? Amino acid. The building block of carbohydrate is the monosaccharides, right? Now the building block of nucleic acid is nucleotide. And we said that nucleotide contains three things: nitrogenous base, pentose sugar, and what? Phosphate group. Now the nitrogenous base can either be a purine or a what? Pyrimidine. Examples of purines are adenine and guanine. While examples of pyrimidines are cytosine, uracil, and what? And thiamine. Good. Now, and we say uracil is only found in what? RNA. While thiamine is only found in? Good. Now, number two, the second thing that can be found in the nucleotide is what? Pentose sugar, right? Pentose. Pentose means 5 carbon sugar. The pentose can either be ribose or what? Deoxyribose. Look at your neighbor and say, don't give up. Don't give up. Oh. Now, ribose, is it found in RNA or DNA? RNA. What's the full meaning of RNA? Ribonucleic acid. Ribonucleic acid. Nucleic acid containing ribose sugar. So this one is found in RNA. That is why it is called ribonucleic acid. Nucleic acid containing ribose sugar. Deoxyribose is found in what? DNA. DNA is what? Deoxyribonucleic acid. Nucleic acid containing deoxyribose sugar. Do we get? Now the third thing that can be found in nucleotide is what? Phosphate group. Good. Phosphate group. If it was in BCH course, we would have seen how all these are interlinked, but nothing is our business here in this course. So in this course, I'm introducing this because we need it in mutation. If you must understand mutation very well, you must understand this. Now, nucleotide contains nitrogenous base, pentose, and phosphate group, right? Good. Now, if you keep this phosphate group aside, what you have, if you remove this phosphate group, is this still a nucleotide? No, right? Because nucleotide must contain these three. So if you keep the phosphate group aside, what you now have is a what? Nucleoside. Because you've kept the phosphate group aside, so you're now having a nucleoside. So nucleoside contains only two things, right? What and what? Nitrogenous base and what? And pentose. You've kept the phosphate group aside. That's why some of you, when your lines, your pickup lines, you now say you are the phosphate in my nucleotide. Without you, I'm just a nucleoside. Because if phosphate group is not there, you are no longer a nucleotide, but you are now a nucleoside. So if you keep the phosphate group aside from a nucleotide, you are nothing but a nucleoside. Do you understand? Please, if I catch you using that pickup line, I should come again, you want to write it. Classification of mutation based on size. Are we there? Number one, point mutation. Number one, point mutation. Number two, chromosomal mutation. Number one, point mutation. Number two, chromosomal mutation. I'll just give you little notes on them. Let's look at the first one, point mutation. This is a replacement or changes in a single base. This is a replacement or changes in a single base. In a single base. There are two types of point mutations. There are two types of point mutations, which are transitions and transversion. Number two, chromosomal mutation. I will explain this. So just write. Number two, chromosomal mutation. These are mutations that affect the chromosome. These are mutations that affect the chromosome. Are we there? These are mutations that affect the chromosome. Paragraph. Examples of point mutation include, or take it as subheading, examples of point mutation. Examples of point mutation. Number one, silent mutation. Silent mutation. Number two, missense mutation. Silent mutation. Number two, missense mutation. Number three, nonsense mutation. Are we there? If you are done with that, just listen up. Let me explain. We all know that mutation is any alteration to the DNA sequence now, right? Good. Now, based on size, okay, based on where they are found, we classify mutation into germline and what? Somatic. Now, based on size, we are classifying mutation into point mutation and what? Now, look at this. Don't cram it. Don't cram it. Let's assume this is the DNA sequence. A, G, C, T, T, A, G, G, C, 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 T. I know some persons are good in cramming, but don't cram this. You can use any of the four letters of the DNA basis. This is the DNA sequence, and we know that any alteration to the DNA sequence is mutation, right? Point mutation is any mutation that affects only one base. These are bases, right? A point mutation is a mutation that affects just one base. Now look at this. If we change this C, right? If we change it to G, like mutation takes place and it changes to T instead of C, that is a point mutation. Mutation that occurs in a single base. Do you understand? That's point mutation. Now we said point mutation can either be transition or what? Now let's recall that our bases can either be what? Bases, nitrogenous bases are either what? Purines or what? Purines or pyrimidines. And we said pyrimidines are what? Adenine and what? Why pyrimidines are what? But this cannot be found in DNA, right? So this one is out. Uracil is out. Now, transition is a type of point mutation in which a purine is changed to a purine or a pyrimidine is changed to a pyrimidine. This is what I mean. Don't give up yet. Don't give up yet. Now let's look at this third base here. This is cytosine, right? Is this a purine or a pyrimidine? A what? Some person stop at the P. It's a pyrimidine, right?
Yeah.
Now, if mutation takes place, and this pyrimidine here, let's say mutation takes place and C is changed to T. T, is it a purine or a pyrimidine?
Yeah.
So a pyrimidine was changed to a pyrimidine, right? In this case, this type of mutation is called trans what? Trans what? Transition. Transition. You're changing purine to purine or pyrimidine to pyrimidine. In this case, pyrimidine was changed to pyrimidine, right? So we call it a what? Transition. Now in this case, adenine, is it a purine or pyrimidine? Purine, right? Now if mutation takes place here and A is changed to G, purine is changed to purine, what type of point mutation is that? Transition, right? Because purine is changing to purine. The same thing is changing to the same group of thing. Transition is just transiting. But cytosine, is it purine or pyrimidine? Pyrimidine, right? If mutation takes place and C is changed to G, G is what? Purine, right? So pyrimidine is changing to purine. In this case, we call it what? Transaction. So in your notes, you can write something like this: transition. Purine to purine or pyrimidine to pyrimidine. Then transaction. You can write something like purine to pyrimidine or pyrimidine to purine. This can go a long way. Are we okay with that? Are we okay with that?
Yes.
So point mutation affects a single base. Now we'll look at example of point mutation, which is always coming out, sickle cell anemia. So using sickle cell anemia as an example, write extensively on. So we'll look at that. Just like I said, we'll look at past questions and how to go about them. Now, chromosomal mutation affects the whole chromosome. Affects the chromosome, not only a base now, but affects the whole chromosome. Do you get that? Now, write this as subheading: frame shift mutation. Frame shift mutation. Is that? No, it's on its own. This is on its own. Yes, completely on its own. I will explain just right. Frame shift mutation. Is that? It's not a mutation. On that side, yes. Did you do all these things in class? Did you do all these things in class? Eh? Now, frame shift mutation. Alright, right. Frame shift mutation. Right. Frame shift mutation is a genetic mutation. Caused by a deletion or insertion caused by a deletion or insertion of a base in a DNA sequence that shifts the way the sequence is read. That shifts the way the sequence is read. That shifts the way the sequence is read. Are we there? Read, like persons of read. Now, Let's look at this. Let's look at this DNA sequence. A, G, C, T, C, T, C, T, C, T, A, T. Good. This is the DNA sequence. I know the DNA sequence, the bases are read in threes, right? 3 bases will give us a codon. Is it not? So these 3 will give us a codon. 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 Is it not? And each codon goes to a particular amino acid. Now, frame shift mutation is any mutation that changes the reading frame. It can be deletion or insertion. Take for example, if this base here is deleted, the reading frame will now become 1, 2, 3, and this, right? You've changed the way the reading frame is supposed to be. This will now code for a different amino acid. This will code for a different amino acid, like that. Do you get? Or let's take for example insertion. You inserted a different base here, say G here. You change the reading frame. Do you get? So frame shift mutation is either insertion or deletion of a base in a reading frame. It will alter the reading frame, and therefore a different protein might be synthesized. Do you understand that? That is frame shift mutation. Any questions so far? Any questions so far? Now, the classwork or the assignment you are giving, that 3 letters, but other stuffs. I will take that assignment, we'll do it together when we are talking about genetic code, because we need the idea there. It's a very easy stuff. Nothing is there. Any questions so far? So if purine is changed to purine, what type of mutation is that? Transition, right? If purine is changed to pyrimidine, is what? Transversion. Examples of mutation in humans. Examples of mutation in humans. Examples of mutation in humans. Number 1, sickle cell anemia. Sickle cell anemia. Sickle cell anemia. Please. This is a genetic disease. This is a genetic disease. This is a genetic disease caused by a mutated version. Caused by a mutated version. Mutated version of the gene that helps in making hemoglobin. Mutated version of the gene that helps in making hemoglobin, a protein that carries oxygen. Is that the mass material? Sorry? On mutation. Okay, if frame shift is under point mutation, it means, now let me clarify this generally. Frame shift can be classified under point mutation if the alteration, if the insertion or deletion is just in one point, it's just in a single base. You get? So if we are removing just one base in the sequence, it's a point mutation, and it's also frame shift because it will affect the reading frame. Is it not? Are you understanding what I'm saying? So frame shift mutation, like if you have something like this, and I remove this base here, I remove this one here, it's a point mutation, right? Because it's affecting just a single base, is it not? It's also a frame shift mutation because if I remove this, it will affect the reading frame, is it not? So frame shift mutation can also be classified under point mutation. But in a case where I remove this and I remove this, deletion, I'm deleting two things. That's frameshift mutation, right? But can no longer be classified under point mutation because it's affecting more than one point now. Do you understand? Okay, that's that. Sickle cell anemia is a genetic disease caused by a mutated version of the gene that helps in making hemoglobin, a protein that carries oxygen in red blood cells. A protein that carries oxygen in red blood cells. This mutation is found in chromosome number 11. One base is mutated. One base is mutated. One base is mutated. In bracket, adenine is changed to thymine. In bracket, adenine is changed to thymine. Adenine is changed to thymine. Full stop. This mistake changes one amino acid. This mistake changes one amino acid added during translation. This mistake changes one amino acid added during translation from glutamic acid to valine. From glutamic acid to valine. We already talked about point mutation. Now, what does it mean when they say base substitution? What does it mean? We are removing one base and putting another base, right? That is base substitution. If in that base substitution we remove a purine and put a purine, what type of base substitution is that? Transition, right? Good. Now, in sickle cell anemia, we know in our blood grouping system, we said we have 4 blood groups, right? Now our genotypes can be what? AA, AS, and what? SS, is it not? So these are the people, sickle cell anemia patients, is it not? Sickle cell patients. So they call them sickler. These people are normal, these people are carriers, and these people are sicklers, right? Good. Now, for those persons that are sicklers, though now we are in an advanced world that they are now bringing out supplements that can help them. Even there was a professor in biochemistry department in Uniport here, Professor Keke, that formulated a supplement he called Siglavit, which they use in treating sickle cell anemia patients, managing the condition. It's sold, like it's late, but it's still sold. Siglavit. Now, most of them they find it hard to thrive, like to survive. But this SS here, what is actually the cause? You know that what oxygen is transported around the body by the help of what? Hemoglobin, right? Hemoglobin is found on the red blood cell. It's a component of the red blood cell. Now this hemoglobin is a tetrameric protein. Let's take it as something like this. It has 4 binding sites where oxygen can bind, so it will transport oxygen around the body. And it's found on the red blood cell. The red blood cell is supposed to be round in shape. But the DNA, on the DNA level now, we have hundreds of bases G to G, G to C to A to like that, hundreds of bases, just like that. And there is a mutation. I'm just taking only one triplet here. There are thousands, hundreds here. Now, if there is a mutation and adenine here is mutated to thymine, A changes to T, there is a problem. That is point mutation, right? Base substitution. Is this translation or transaction? Trans what? Transition or transaction? Transaction, right? This is a purine, right? Changing to a pyrimidine. So it's a transaction, is it not? Now, A is what? Is it a purine or a pyrimidine? And A is changing to T here, right? C, is it a purine or pyrimidine? So purine is changing to pyrimidine. Transition or transaction? Now, this is the only thing happening. No, there are hundreds, but just in one position. A changes to T. And so if we write the complement now, the complement of G will be what? A will be what? G will be what? Down by market. Now, look at the mutated sequence now. G will be what? C will be what? G will be what? Now, this is for the complement. Now, let's look at the RNA. The RNA now. This is G, right? This is A. A can also be found in RNA. G can also be found in RNA. Is it not? Coming here, G can be found in RNA. But C cannot be found in RNA, right? Instead of C, what do we have? U. Remember, you are seeing one in RNA in place of what? Thymine, right? G can be found. Now, this GAG and GUG. This GAG goes for glutamic acid and amino acid, while GUG goes for the amino acid valine. Valine. As you can see, the amino acid produced have been changed. Out of hundreds of bases, adenine was changed to thymine, and now glutamic acid have been changed to what? Valine. Just this one change. Red blood cell cannot be round again. It becomes sickle shape. Sickle is like that thing that harvester, that thing they used to harvest, or half moon shape. You see that you call it sickle shape or half moon shape. Half moon shape. No longer round. And so you cannot transport oxygen around the body effectively again. That's what is happening in sickle cell anemia patient. That critical condition is just a mutation in one base. What base is changing to what? Adenine is changing to what? Thymine, right? And what amino acid is produced? Valine in place of glutamic acid. And it results to that serious problem. Back to the past question now. The question says, from the what? Give detailed description from what? Thank you very much. So we said, give a detailed description of how the effect of a base substitution can be traced from DNA level to the level of the whole organism, using sickle cell anemia as an example. In answering this question, what I've said is what you say. Now, in answering this question, you can start with this. Write this on the book, on the notes for them. You can start with that. DNA, GAG, mutated to what? GTG. Don't worry, don't bother about cramming it. You get it in your head before you leave this class. Now, the complement, you know how to write complement already, right? Then the RNA. Then the proteins, the amino acids produced. Good. Now, after writing this for them, he said, from the illustration above, on the DNA level, adenine was mutated to what? Thymine. which led to the production of valine instead of what? Instead of glutamic acid, right? On the level of the whole organism now, the red blood cell, because due to this mutation, can no longer be run, becomes what shape? Sickle shape, and can no longer transport oxygen effectively again, leading to the condition sickle cell anemia. So basically, don't cramp. Answering that question, the answer to that question is written in this book here. I said at the DNA level, the base adenine is substituted for, is written here, clearly written, but I don't encourage cramming. That's why I didn't even bother dictating that part. Understand it, write it in your own terms. Do you understand what is happening here? Now, I'll take it again. For sickle cell anemia, what base? Remember, we said mutation is any alteration to the DNA sequence, right? What base is altered in sickle cell anemia? Adenine is substituted to what? Thymine, right? So instead of adenine, where you're supposed to have adenine, just 1.2, you now have thymine. And because of that, adenine changing to thymine, if you are writing the RNA, GAG, G cannot be found in RNA, U. So U is found G, GUG. Now this is a codon that codes for glutamic acid, and this is a codon that codes for valine. So in the amino acid chain, instead of saying glutamic acid there, what you will now see is what? Valine. Only that change at that point will affect the shape of the red blood cell. And so it becomes sickle shape, half moon shape, and can no longer transport oxygen effectively again. That is the sickle cell anemia condition. Do you understand? Do you understand? If you forget everything we said today, don't forget that polymorphism is the what? Variation between normally healthy individuals. Don't also forget that medical genetics is the what? Branch of genetics that deals with what? Diagnosis and what? Management of hereditary disorders. Don't also forget that the universal recipient is what? AB positive, and the universal donor is what? O negative. Don't also forget that mutation is what? Alteration in the what? DNA sequence of an organism. And classification of mutation based on where they are formed, we classify mutation into germline and what? Somatic. Which one can be passed from parents to offspring? And based on size, we classify mutation into what? Point mutation and what? Chromosomal mutation. Now, point mutation, we said it can be transition or what? Now, we said also that an example of point mutation is sickle cell anemia, right? Good. And in sickle cell anemia, we said A is changed to what? So when we're supposed to have GAG, what are we having now? When we're supposed to have GAG, we're having what? GTG, right? Now, when we get to the RNA level, we'll see why we are transcribing it to RNA, why we are converting it to RNA. The genetic code, what we use in the genetic code is actually the RNA. The RNA code. We'll see that. So that will make it clearer when we get to that point. Now, GAG and GUG. So in place of glutamic acid, what are you now having? Valine, right? That is sickle cell anemia. Are we good to go? Next one. Cystic fibrosis. Number 2. We are looking at examples of point mutation, is it not? Number 2, cystic fibrosis. Do you call it cystic fibrosis? Cystic fibrosis. This occurs one in every 3,000 births. This occurs 1 in every 3,000 births. Are we there? The mutation occurs in a gene located on chromosome 7. That of sickle cell anemia occurs on chromosome what? 11. The mutation occurs in a gene located on chromosome 7. This occurs 1 in every 3,000 births. Full stop. The mutation occurs in a gene located on chromosome 7. In a gene located on chromosome 7. Full stop. Persons affected with cystic fibrosis. Persons affected with cystic fibrosis. Persons affected with cystic fibrosis produce thick, sticky mucus. Persons affected with cystic fibrosis produce thick, sticky mucus in their lungs. In their lungs, comma, in their lungs, comma, intestines and pancreas. In their lungs, intestines and pancreas. Persons affected with cystic fibrosis produce thick, sticky mucus in their lungs. In their lungs, comma, intestines and pancreas. And pancreas. Full stop. The buildup of thick mucus blocks breathing passages. The buildup of thick mucus blocks breathing passages. Are you lost? Or you're not writing? You don't give up. Don't give up. Oh. You don't give up too. Where are you? Don't fool. Wow. The buildup of thick mucus blocks breathing passages. Are we there? And makes waste elimination difficult. And makes waste elimination difficult. And makes waste elimination difficult. Are we there? Number 3. Down syndrome. In bracket, trisomy 21. Do you still remember this genetics 1? Number 4, Cri-du-Chat syndrome. If you attended my FSD 202 class, I don't expect you to have forgotten anything there. You forgotten? Okay. Number 5, Klinefelter syndrome. Number 6, triple X syndrome. Number 7, monosomy. In bracket, Turner syndrome. Turner syndrome. Number 8, phenylketonuria. Number 9, albinism. Number 10, Lesch-Nyhan syndrome. So all these names are familiar names, right? Above Lesch-Nyhan syndrome, didn't we do it? Oh, okay, BCH. BCH. Actually, we saw it in metabolism of nucleotides. Lesch-Nyhan syndrome. NYHAN syndrome. All right. Now, cystic fibrosis. We said it affects chromosome number what? 7, right? Now, in cystic fibrotic patients, they find it difficult to breathe, first of all. Now, accumulation of thick, sticky mucus, very thick. And this will block breathing passage. You know, your lungs. Let's take something like this as your lungs. Not mine, obviously not mine. Let's take something like this as your lungs. How many do you have? You have 2, right? Remember, you have 2 tubes here, right? One leads to your stomach and one leads to your lungs, is it not? And lungs is involved in what? In breathing, right? Taking in and out of air, is it not? Good. Now, if there is a thick mucus here, sticky, that is blocking the respiratory tract, that is blocking the pathway where air was supposed to pass, you find it difficult to breathe and you cannot remove waste. Carbon dioxide is supposed to go out, right? But this is blocking this. This is blocking this. So that's the case of cystic fibrosis, though it's actually rare. We say it occurs 1 out of 3,000 births. Now, other examples of point mutation. Down syndrome. I won't talk much on this because we've done them before. 202. Down syndrome, we also call it what? Trisomy what? And what is trisomy? Having an extra copy. You know, there are supposed to be 222, right? Having an extra copy of a particular chromosome is called trisomy, right? So trisomy 21, instead of having 2, we have an extra copy. This trisomy 21. It happens on chromosome number 21. So trisomy, you are adding an extra copy of a chromosome. Chromosomes are always in pairs now. 2, 2, 2, 2, 2, 2. But in trisomy, you are adding 1 extra, making it 3. It's actually a disorder. So trisomy, Down syndrome. Now, Cri-du-Chat syndrome, we looked at it, we said it affects children and as a result of malformed larynx, right? And so since their larynx is malformed, they cry like what? Cats. That is how the word comes about. Cri-du-Chat. Cry like cats. They cry like their larynx is malformed. Malformed larynx. It's not well formed. So when they want to cry or when they want to talk, their voice is not, sounds like that of a cat. And so they call it Cri-du-Chat syndrome. And we say it affects the short arm of chromosome number what? 5. Short arm of chromosome number 5. Now, Klinefelter syndrome. Klinefelter syndrome, you can see it in something like 47XXY. 47XXY, 48XXXY, like that, it continues. Now, Klinefelter syndrome, what is the karyotype of male? That of a male, is it XY or XX? That of female is what? Does female have Y chromosome? They can never in their lifetime. They can never. So it's only males that have what? The Y chromosomes, right? Yes. It's only males that have Y chromosome. Good. So at least, if they want to say what we have, what they are bragging anywhere, tell them they can never have Y chromosome. They are not here. Alright, so now the Klinefelter syndrome, does it affect male or females? Because females cannot have Y chromosome. So this affects only males. But now the Klinefelter syndrome, the male affected, they are normally very tall, and they have feminine body, they have breasts, and they have small genitalia, small penis. In FSB, who said that child? In FSB 202, I gave an instance of a case where they saw somebody like that. and they were beating the person. That's FSD 202. If you are not there, it concerns you. Alright, so that's Klinefelter syndrome. They have multiple X. It continues, can be 49 XXXX, while like that. So it's just like this in this order. Very tall. Please, I'm not tall, I'm average. Alright, so the triple X syndrome affects only which people? Only females, right? So they have an extra copy of the X chromosome. Can it be passed on to their offspring? Can it? Because it affects the sex chromosome, right? So it can be passed on to the offspring. Remember, germline mutation can be passed on to the offspring. Now, XXX, they have an extra copy of the X chromosome. That's for the female, the triple X. Then monosomy. Monosomy is also called what? So now in place of, you are supposed to have 2, 2, 2, 2, right? But one is missing, so you have only one. That's monosomy. Trisomy, you have an extra, making it 3. Monosomy, one is missing, so you have only one copy of a particular chromosome. You don't have 2 copies. That's monosomy. Turner syndrome. Now, phenylketonuria, we'll still come back, yes. This for female. Now them. Phenylketonuria is actually a defect in amino acid metabolism, which we are going to look at. I'm going to look at this. And for those of you that have phenylketonuria and alkatonuria, you can actually detect it yourself. When we get to that topic, which will likely be the next topic, you will detect it. As you go home, I'll give you as assignment to go and detect if you have alkatonuria or phenylketonuria. You can detect it without testing anything. I'll show you that. Albinism. Albinism. Now, if this is also let me not explain much here because it's also a defect in amino acid metabolism. Do you know that? The albinism is a defect in amino acid metabolism. Yes, it's a defect in thyroxine metabolism. What gives you your pigment? This pigment? Melanin, right? But in albinism, there's an enzyme called thyroxinase. Thyroxinase is absent or defective. We'll talk about that when we get to that. Leishman syndrome is actually a defect in nucleic acid metabolism. So these are examples of point mutation, just single point mutation, single point mutation, and that's it. So nucleic acid metabolism. It's a defect in nucleic acid metabolism.